Hello, everyone. Our project is the RT-LAMP microfluidic rapid detection of SARS-CoV-2. So a little bit background on our introduction to our project is I'm sure we're all too familiar with this coronavirus, but it has caused over 1 million deaths this past year, and I'm sure this number is even higher at the moment, even with vaccines. And this COVID-19 is caused by the coronavirus SARS-CoV-2. Um, Thus, the need for a quick, at-home, low-cost widespread test is really important, especially emphasis on the widespread and frequent testing. Current methods involve polymerase chain reaction, which must be done in the lab and is time and money intensive, which can also be very invasive going through the patient's nose or throat. Our test proposed to use saliva. This is both non-invasive and yield more accurate results. Through saliva, our test can detect COVID-19's RNA polymerase, as seen to the right. And next, Sophia will discuss the relevance of the project to you all.
Thank you, Kaylee. The motivation of this project is that Boise State University needs to be able to offer rapid testing for all students and faculty members in order to increase in-person campus activities, increase the safety limit and spread of COVID-19 to our community. And then beyond just Boise, Idaho, we want to make an impact anywhere that we can. And these include rural communities that may not have access to testing facilities. Additionally, these tests would be able to provide reliable results prior to symptoms and additional exposure of COVID-19. And next, I will pass it off to Ashlyn, where she will give us the background of this project.
Thank you, Sophia. So RNA is a key regulatory player in the cell and results from the transcription of DNA, which is then translated into proteins that is used for bodily functions. SARS-CoV-2 is an RNA virus that uses surface-spiked proteins to enter cells to create viral proteins that interfere with the immune system. So loop-mediated isothermal amplification, LAMP, is a type of test where it's able to amplify a targeted DNA sequence. Reverse transcription loop-mediated isothermal amplification, or RT-LAMP, has been adapted from LAMP in order to detect viral RNA through the addition of a heat-stable reverse transcriptase. So RT-LAMP has the advantages of being cost-effective, only limited resources needed, and the reaction is run at a single temperature in just one step. So for paper microfluidics, these are typically done on cellulose paper. They're usually wax printed, and their advantages are they're affordable, user-friendly, sensitive, rapid, less environmental impact, and mass producible. And here's an example of RT-LAMP on a paper microfluidic device.
For our technical approach, we designed a microfluidic channel and printed it on Whatman 4 cellulose paper. It was printed with wax ink on a Xerox printer. Then we had to reflow the wax through the depth of the paper by placing it on a hot plate for 5 minutes. This prevents liquid from leaking out of the channel as it travels. Once we completed the device, we had to test the capillary action to see how well liquid would flow through the channel. We dropped about 1 milliliter of colored water into the reception end of the channel and watched the water flow through. Once we had a device that would flow, we tested RT-LAMP. This was performed by placing 2 microliters of sample in the outlet ends of 2 channels and let them dry out. One was a positive control and the other was a negative control. Then 23 microliters of RT-LAMP reaction mix was placed onto the reception ends. The channel was then heated on a hot plate for 30 minutes at 65 degrees C. When the channels were taken off the hot plate, we looked to see if the channels had changed color. Now I'll hand it off to Maddie for results.
Thank you, Zoe. Here are results of the wax printing that was done with help from the Macromolecular Sciences Laboratory for our paper microfluidic device. On our printed devices shown here, you can see that we attempted 2 different options of designing our microfluidic devices. First, we created a prototype on SolidWorks. These photos are shown up at the top right. And you can see that in the SolidWorks design, the wax was printed onto the channels, which made this attempt unsuccessful since the liquid wasn't able to flow through the hydrophobic wax, which you can see in the kind of grainy on the picture. After this, we created a prototype on PowerPoint. The wax printed from PowerPoint did not print onto the channel, which means that we had a successful channel for liquid to flow through. As for the rest of our results, please attend our Senior Design Showcase, which is on Zoom on April 27th from 4 to 6 Mountain Time. Information is on Boise State's Cohen website. Quick thank you to all of our sponsors and the laboratories that were able to help us with prototyping and testing. You all made this possible. Thank you.